Far beyond the Great Barrier Reef, a camera crossed a dark seafloor that had rarely been seen. A large shark moved through the lights. On the deck above, scientists were bringing up animals that did not match the scientific record. The first announcement counted more than 110 new-to-science fish and invertebrates. The number has already increased, and researchers expect it may eventually pass 200. This is the story of the Coral Sea Frontier Voyage, where it went, how the animals were collected, what the new species look like, and why finding an unfamiliar creature is only the beginning of the scientific process. There is an important distinction. These animals have been identified as new to science, but most have not yet been formally described or given species names. The evidence is strong enough to recognize that they do not match known species. The discoveries came from the Coral Sea Marine Park, east of Queensland, and beyond the Great Barrier Reef. This official bathymetric map shows the continent at left and the enormous field of plateaus, basins, reefs, and underwater mountains to the east. At almost 990,000 square kilometers, it is the largest marine park in Australian waters. It protects 34 vast reef areas and 67 cays and islets, while its deepest parts descend to about 6,000 meters. The voyage concentrated on the Marion Plateau, the Ken Plateau, and the northern Tasmantid Seamount chain. These extinct volcanic peaks rise more than 3 kilometers from the surrounding seabed, creating isolated habitats at many different depths. Research vessel Investigator left Brisbane on the 10th of October 2025 and returned on the 14th of November. The 35-day expedition traveled as far as Mellish Reef, roughly 1,000 kilometers from the Queensland coast. The aim was not simply to collect unusual animals. Researchers wanted to compare biodiversity across seamounts, atolls, plateaus, and continental slopes, test how isolated populations are connected, and learn which survey methods give the most reliable picture. The team successfully surveyed 61 sites, 6 more than originally planned. Those sites ranged from about 200 meters to 3,600 meters deep, crossing several distinct environmental zones. Investigators' multi-beam echosounders mapped the seabed while the ship moved. The voyage completed the first full mapping of Mellish Seamount and revealed a complex canyon system on the Marion Plateau. At the shallow end of the survey, faint sunlight may still influence life. Deeper down, daylight disappears completely. By 3,000 meters, every organism lives under crushing pressure in cold water, with food arriving irregularly from above or along the seafloor. The deep sea is not one uniform habitat. A rocky peak, a sediment plain, a steep canyon wall, and a coral-covered ledge can support very different communities, even when they are separated by only a few kilometers. Before sampling, mapping helped the crew identify safe and scientifically useful locations. It reduced the chance of placing heavy equipment on steep or fragile ground and gave every specimen a much clearer environmental context. A deep-towed camera recorded animals in place without removing them. The system can operate to about 3,900 meters, sending images back to the ship so researchers can observe habitat, behavior, and abundance. Camera observations are non-extractive, which makes them valuable for sensitive habitats. But a photograph cannot always reveal the microscopic structures, internal anatomy, or genetic differences needed to recognize a new species. A CTD package measured conductivity, temperature, and depth while collecting seawater. Those readings show the physical conditions surrounding each biological community. Water samples were also searched for environmental DNA, tiny traces shed as mucus, scales, cells, and waste. This can reveal organisms that escape the camera and the nets, although it depends on having reliable genetic references. Trawls and sleds provided the physical specimens needed for detailed identification. These methods were used carefully at selected sites, after mapping and camera work had helped the team assess the bottom. An epibenthic sled skims the seafloor and gathers small animals living on or just above it. When it returns, the contents may look like mud, rubble, and fragments, but hidden among them can be hundreds of species. As soon as the catch reached the deck, the real race began. Living animals had to be photographed, sorted, labeled, and sampled before color faded or delicate structures changed. Each label connects a specimen to its station, depth, time, gear, and environmental data. Without that chain of information, an unusual animal loses much of its scientific value. Across the voyage, the team collected more than 6,000 fishes and more than 80,000 invertebrates, together with thousands of voucher specimens for museums and research collections. A voucher specimen is preserved reference material. Future researchers can re-examine it, compare it with later finds, sample its tissue, or test a new idea that did not exist when the voyage took place. After the ship returned, CSIRO and the Ocean Census brought specialists together in a series of Australian workshops. Fish experts, sponge experts, worm experts, and many others worked through the material group by group. CSIRO described these as likely the largest taxonomic workshops for marine animals ever held in Australia. Hundreds of specimens could be compared side by side, while international knowledge was concentrated in the same room. The first line of evidence is morphology: body shape, teeth, spines, fin positions, scales, reproductive structures, and microscopic details. A tiny feature can separate two species that otherwise look nearly identical. Genetic sequencing supplies another line of evidence. It can expose cryptic species, test relationships, and link fragments or life stages that would be difficult to match by appearance alone. When morphology, genetics, and the scientific literature all indicate that an animal is distinct, researchers can identify it as new to science. Formal description comes later, after exhaustive comparison and peer-reviewed publication. One of the most striking candidates is a deepwater cat shark in the genus Aprosturus. Shark specialist Dr. Will White recognized that this specimen did not match any described member of the genus. Its long, dark body, small fins, and tapering tail are documented against a scale, with the collection label preserved in the frame. Those proportions and measurements will become part of the formal description. For now, it has a working identification rather than a species name. Its behavior, diet, range, and population size remain largely unknown, so the specimen should not be used to invent a complete life story. The first ray candidate belongs to Dipterus, a genus of skates. The voyage material calls attention to its triangular tail and records it only to genus level, while the detailed comparison continues. Skates are rays, so both words may appear in reports. In this dorsal view, the broad disc is formed by enlarged pectoral fins, while the long tail extends behind it. A second ray candidate was placed in the genus Urolophus. The identification includes the cautious notation CF papilio, signaling resemblance to a known form without claiming that the two are the same species. Its rounded disc and relatively short tail create a very different outline from the skate. External shape is useful, but specialists must also compare structures and genetics before a name can be published. Dr. White also identified a new chimera, sometimes called a ghost shark. No confirmed photograph of that new candidate was supplied. The animal shown here is a known species from the voyage, included only to show the general body form of a chimera. The new fish were not limited to sharks and rays. This colorful specimen is a new-to-science wrasse in the genus Chirodon, the group commonly known as tuskfishes. Among the invertebrates is a brittle star in the genus Ophiozunella. Several specimens were photographed together, giving taxonomists a chance to compare natural variation rather than relying on one individual. Brittle stars are relatives of sea stars, but their slender, flexible arms are sharply separated from the central disk. Small differences in plates, spines, and arm structure can be crucial for identification. This jellyfish was identified by Australian Museum specialist Dr. Claire Rowe. Its transparent bell and internal structures were photographed fresh, before preservation altered its appearance. Jellyfish are commonly fixed in formalin, so their form is preserved, but formalin makes later DNA extraction difficult. During the voyage, researchers can take tissue first and then preserve the specimen for morphological study. A small gastropod can be just as scientifically important as a shark. Shell shape, sculpture, aperture, and internal anatomy all carry taxonomic information, while DNA can test whether similar-looking shells belong to separate species. These segmented polychaete worms were examined by specialist Elena Kuprianova. What looks like a simple pale thread can carry a complex arrangement of bristles, appendages, and body segments. A second tray shows a different set of curved specimens. Sorting many individuals together helps reveal which differences are consistent, which change with age, and which are simply variation within one species. The voyage also produced new sponge candidates. Sponge taxonomy may depend on microscopic skeletal elements called spicules, the arrangement of tissues, and genetic evidence, not just the overall shape seen in a photograph. Another sponge sample appears almost fragmentary beside its labels. That is a reminder that scientific significance is not measured by size or beauty. A small piece can preserve diagnostic structures and genetic material. CSIRO also reported new species among crabs and sea anemones, with more work continuing across jellyfish, sponges, worms, and other groups. The public images show only a small sample of the full discovery. Not every remarkable animal collected was new. The media archive deliberately includes known species as well. Keeping that distinction visible prevents an exciting expedition from turning into a misleading list. The known finds included a fierce-looking deep-sea lizardfish, batfishes, an octopus, anemones, corals, ostracods, and squat lobsters. They help describe the community around the new species and expand records familiar animals live. One of the camera highlights was a rare sand tiger shark, Odontaspis ferox, filmed at Mellish Seamount at a depth of 340 meters. This shark was not a new species. It is a deepwater relative of the better-known grey nurse shark. Seeing it alive in place contributes information that a preserved specimen cannot provide: posture, movement, habitat, and the other animals around it. Why did one voyage find so much novelty? Part of the answer is simple. This region is enormous, deep, and poorly sampled, but the shape of the landscape also matters. Seamounts can behave like underwater islands. Populations on one peak may be separated from relatives on another by deep basins, while currents can sometimes carry larvae across great distances. The voyage was designed to test those patterns rather than assume them. At Mellish Seamount, The team also found a previously unrecorded, apparently pristine mesophotic coral ecosystem. Mesophotic means the low-light zone below most shallow reefs, linking familiar coral habitats with the deeper frontier. The headline figure of more than 110 came from the first April announcement. The later voyage summary from CSIRO reports more than 120 species already identified as new to science. The count is expected to exceed 200 as more cryptic species are recognized. That is an estimate, not a promise, and some candidates may change as experts compare additional material. Cryptic species are especially difficult. Two animals may look nearly identical but represent long-separated genetic lineages. The reverse can also happen. Individuals that look different may prove to be age, sex, or color variants of one species. To formally describe a species, scientists must select type material, measure and illustrate it, diagnose how it differs, compare museum collections and published records, choose a valid name, and publish the work for scrutiny. That work can take years. CSIRO notes that marine species wait an average of 13.5 years in collections before formal description. Investigators' first 12 years produced 179 published species descriptions, while more than 1,500 candidates were still waiting. Coral sea specimens are now lodged in collections around Australia, including the Australian National Fish Collection at CSIRO and state museums. These institutions keep the evidence available for decades, not just for the scientists who join the voyage. The records also feed open biodiversity systems. Workshop specialists entered data into the Ocean Census Biodiversity Data Platform, while voyage information can later support national and international repositories. The research has a practical purpose. Parks Australia needs reliable evidence to manage fishing, conservation zones, and environmental change. The voyage was designed to provide data for the next Coral Sea Marine Park Management Plan. A baseline is most valuable before a major change occurs. If future surveys repeat these methods, managers can compare communities through time and distinguish natural variation from sustained shifts in distribution, abundance, or habitat condition. More than 100 new species from one expedition does not mean the ocean suddenly changed. It shows how much life was already there, beyond the reach of ordinary observation and outside the scientific catalogue. The Coral Sea Frontier Voyage turned a remote seafloor into maps, images, specimens, and testable evidence. The names will come slowly. The larger discovery is already clear. Australia's deep sea is far more diverse than the existing record can show.